In this video, we will discuss carbon dating. So, what is it? Carbon dating is a scientific method used for determining the age of organic materials younger than 60,000 years old. So, no metals and no dinosaurs. But how does it work? Well, first we need to understand cosmic rays. These are positively or negatively charged high-energy particles from outside of our solar system, which move insanely fast, as in almost at the speed of light. They vary in their makeup. Around 90% of their nuclei are hydrogen, aka protons, 9% helium, aka alpha particles, and the last 1% is made up of other elements. Whilst these cosmic rays hit the Earth, they produce neutrons, which then nuclearly react with the nitrogen found in the stratosphere to form 14C, or carbon-14. The neutron bumps off one of the protons of nitrogen and replaces it with itself, meaning there are 6 protons and 7 neutrons, which is carbon-14. The extra proton that was lost is emitted in the form of hydrogen.
These atoms can also be formed as a result of atmospheric nuclear testing done in the 1950s and 1960s. The nuclear bombs brought lots of thermal neutrons, which then reacted with nitrogen in the atmosphere. The result was a massive increase of C14 levels. This is also called the artificial radiocarbon or bomb carbon. After C14 is formed, it very readily oxidizes to form carbon monoxide, which later turns into carbon dioxide. This inert gas is found in the stratosphere and troposphere, yet also on the surface of the Earth. Carbon dioxide 14 is an isotope of carbon dioxide, and it enters into what is known as the global carbon cycle, which is just a cycle of carbon being reused. It is absorbed by different organisms, and is then released through various processes, such as burning fossil fuels, decay, etc. So, when it enters this cycle, it is absorbed by humans, animals, plants, etc., through their intake of carbon dioxide. When these living organisms die, they stop the intake of C14, and its levels slowly decrease in the organism. The C14 decays back to nitrogen.
Based on how much C14 is left in the organism, scientists can determine its age. There are 3 main methods for this. The first is called gas proportional counting. This technique first converts the C14 into CO2 gas, and then the number of beta particles emitted is counted, since those particles are the outcome of radiocarbon decay. It is measured in gas proportional counters. The second method is called liquid scintillation. The scintillator is added to the sample in the liquid state, and when it reacts with a beta particle, it shines. A count is made only when the 2 photomultipliers between which the vial with the sample is positioned note the light.
The last method, also believed to be the most efficient, faster, and most modern, is the accelerator mass spectrometry, also known as AMS. It measures the amount of carbon-14 directly, instead of its product, and requires only very small samples. So, how does it work? Living organisms not only absorb the unstable C14 isotope, but also the stable C12 and C13 isotopes, in a similar ratio of how they were naturally. These are not radioactive, and so scientists can compare how much C14 is left relative to the amount of C12 in the material. So, no beta particles are counted, but instead the carbon themselves, and the proportion of the isotopes. After 5,730 years from the death of the organism, 50% of the carbon-14 atoms have decayed, whilst 11,460 after its death, 1/4 of the carbon atoms are left. This means that the lifespan of C14 in an organism is quite short, and can only be used to determine the organism's age up to 60,000 years ago. When is carbon dating used? Carbon dating is mainly used by archaeologists to find the age of trees, plants, animal remains, ancient artifacts made out of wood or leather, artworks, and fossils. It can be used in studying environmental pollution and even tracking the movements of animals or plants. What are the pros of it? The discovery of carbon dating had a great effect on geologists, paleontologists, and archaeologists. This is because before the use of carbon dating, they would have used relative dating, which is when an artifact, for example, is determined on how old it is based on its relations to other artifacts. This would only, however, verify if something is older or younger than other artifacts, and not the year or time period it was made in. Carbon dating, on the other hand, falls under absolute dating, meaning that it can tell us the exact year that an object was used or something had died. Carbon dating can date an artifact specifically that is less than 60,000 years old. Carbon dating can help us date how old living organisms are, since it's based on the contents of carbon-14 in organic material. So, it can't date pottery or other items that did not contain carbon-14. However, any living organisms that are related to the non-living artifacts can be dated, hence allowing the dating of inanimate objects. In other words, carbon dating can help dating a plethora of items and artifacts regardless of if it's living or not. Some examples are seeds, textiles/fabrics, and bones. As mentioned before, carbon dating can be done through AMS dating, which is a more recent technique used. Before the use of AMS, carbon dating required quite an amount of substance to get accurate results and measurements of carbon-14. The use of AMS, however, allows us to use much less material of an object we are trying to date. Currently, we are able to use a sample size of a single P. This ultimately preserves a large amount of artifact being carbon dated. The use of carbon dating has helped us interpret when older climate changes took place far more accurately. For example, scientists used to believe that the ice age in North America had roughly happened 25,000 years ago, but the use of carbon dating was able to actually show that the ice age was actually only about 11,000 years ago by using samples of trees that were found under glacial ice. This eventually affected many climate chronologies and the scientists who spent years researching the chronology of climate change in the past.
What are the limitations and cons to the method? To start off, carbon dating is useless after 50,000 or 60,000 years because so little carbon-14 is left on the sample that calculating age accurately is no longer possible. Carbon dating also cannot be used on inorganic matter, with few exceptions if it came in contact with organic matter, for example. Examples of matter that could be tested, however, are wood, cloth, bones, and fossils.
Carbon dating is also affected by the temperature the organism was buried in. If it was buried in a warm environment, the C14 would decay quicker, making the dating unreliable. There's also the problem of changing amounts of C14. Since the 1950s, for example, the levels of C14 exceedingly increased. These changing levels mean the basing levels of C14 we would expect to find in something very old on today's standard would be inaccurate. Also, the effect of cosmic rays was also changed due to the changes in Earth's magnetic field and solar cycles. Thankfully, there are ways to fix that problem, by looking at the tree rings, for example. Every year a tree grows, it produces one exoring, which indicates the level of C14 for that year. Scientists can also determine the amount of C14 of that time period by looking at the corals and their C14 and C12 levels to be able to study samples from up to 50,000 years ago. Carbon dating also decreases in accuracy the older the sample gets. For a 50,000-year-old sample, the accuracy varies with around 100 years, whilst a 10,000-year-old sample only varies between 10 years. Additionally, a sample can be contaminated by other matter, such as soil, which can throw the accuracy of the dating. One of the major cons is the high cost of carbon testing. An average testing costs $250, and an AMS apparatus costs $2 to $3 million US. As we can see, there aren't many cons to C14 or carbon dating, meaning it is a fine and fairly accurate method to use, although it is expensive and has its limitations. However, its health, environmental, and economical impacts are scarce, since the process of formation of C14 occurs naturally and carbon dating does not interfere with it. What are the dangers of carbon dating? Since the human body constantly comes in contact with radiation and even absorbs C14 regularly, it is not dangerous in small doses, only if it goes over the limit, which is 2 MCI for inhalation and ingestion yearly. C14 emits beta particles at a low energy, which means that even an external exposure to large amounts of it is not a big threat. It scarcely penetrates the outer dead layer of the skin. However, there is a bigger risk if someone comes in contact with it internally, through ingestion or inhalation, which means that there are some safety precautions when handling it in a lab, such as wearing safety goggles and wearing gloves and changing them frequently. If clothing gets contaminated, it should be disposed of. If there are any spills, it should be properly cleaned. There have been concerns about C14 being present in the food and air, especially in higher amounts because of the bomb testing in 1950s and 1960s. Yet even then, one could eat 200 kilograms of vegetables per day, as the National Library of Medicine puts it, meaning it is not that dangerous for humans to ingest or inhale in regular amounts.